This is TWIS. This Week in Science, episode number 946, recorded on Wednesday, October 4th, 2023. Science is always Nobel. I'm Dr. Kiki, and tonight we will fill your heads with mushrooms, mistakes, and purring. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. The following program contains dangerous information related to the nature of reality. Within the words, terminologies, and phrases you are likely to encounter here are the tools humans use to separate fiction from fact. The difference is real. As different as a sci-fi fantasy and actual space exploration, a children's story, and a study into childhood illness, any cultural mythology, and every molecule that can cure a disease. The information is dangerous because the human mind is a collection of fantasy, mythology, and storytelling. Without them, it is unclear who or what a human being is. Assumptions, opinions, beliefs, and biases are the underlying conditions blurring the human mind, responsible for our unfocused actions and responses. They are seductive sedatives, a blissful blindfold, a well-intended illusion. And they have poisoned men's souls, barricaded the world with hate, goose-stepped us into misery and bloodshed again and again. Assumptions, opinions, beliefs, and biases. What can be seen without them? What can be learned without them? What can be done without them is science. And there's no better place to start seeing the world for what it really is than This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
And a good science to you too, Justin. Welcome to another episode of This Week in Science. And thank you all for joining us for yet another adventure into the land of curiosity, inquiry, science.
Yeah.
Oh, yes. Yes, we are here. Let's see. We got a lot of stuff going on this week. It's been a big week. I have stories about the Nobel Prize Awards, a new organelle.
Again? I felt like we just did that last week.
I know. It's like it happens over and over again, and I feel yeah. Anyway, I got a new organelle. Cats.
A new organ? Wait, a new organ? You just skipped past, like you went like, there's cats. There's a new organelle.
Yep.
Wow.
Yeah, and there are many questions about it, so it's an interesting conversation. Mushrooms. We like mushrooms. I got mushrooms and memories. Memories.
Whoa,
what do you have
whoa.
for us?
I've got the meaning of Stonehenge.
It has a meaning?
Yeah.
Oh. I
That
thought it
henge
was just a
is
bunch of rocks. People are like, dude.
Well, okay. Hang on. No spoilers,
Okay.
but it does have to do with rocks.
Okay.
Story. Yeah.
Totally rocks.
I have a chilling tale of snowflies, self-amputation, survival, and certain death.
Certain death.
Bedtime math lessons. Maybe something that's in the future. Future children will rejoice.
Well,
the opportunity.
counting sheep. Instead of counting sheep, it's math sheep. Okay.
Sort of multiplying sheep right
Yeah,
before
okay.
bed. Yeah, forget counting them. That's the old way. From now on, we're doing a multiplication table of sheep. And this story, I don't know. It's a sort of a history study. of medieval murder
Oh.
that centers around Oxford University.
This sounds like it could be its own podcast, like a whole story.
I'm sure. No, it is. There's
dig.
stories within stories.
Yeah.
But it makes Oxford look like Chirac. Like the most
Murderhenge?
violent.
It look like a bloody mess. Like
medieval university. You know how we have, we'll get into this story later.
Yeah, we'll get into it.
You know how we have like clubs on campus? You can join like different like interest clubs, and you can join different sports
but there's the secret societies too. Like the
secret societies, but you're still all in the same campus, right?
Yeah.
Well, back then when the world was smaller, like being part of the chess club versus like the Go club or whatever, when everybody had like sidearm knives.
Oh, geez.
Anyway.
Can't wait.
People weren't good at arguing, I don't think. Anyway, we'll
they
get into
were
that
good
one.
at killing each other. All right, let's talk about it. Oh my goodness. There is mystery and mayhem afoot. All right.
Murder
most foul. Yes, let's talk about all of it. It is spoopy month, so it totally fits. As we jump into the show here tonight, I would love for all of you who are watching right now to share the show with whomever you think needs to hear it, and also like and subscribe. If you're not yet subscribed to us, you can find us as a podcast, most places that podcasts are found. You can also find us live streaming on YouTube, Twitch, and Facebook, 8 p.m. Pacific time-ish on Wednesday nights. Our website is TWIS.org if you are interested in our show notes, links, and other exciting bits from the show. Also, we have links to help you subscribe. So if anything, TWIS.org is the thing that you really need to remember. But now is the time for science. You ready?
Oh, yeah.
Okay. Well, we started last week's show with the Ig Nobels, so we would be remiss if we didn't start this week's show with the Nobels.
That's why I was confused. I thought those were the Nobel Prizes. That's why I was so confused last week. Because I was like, I didn't think there was better. Just like last week, there was
pretty
more
good science.
Impactful research than turning, what is it, necrobotics. I think there's more useful stuff.
Like, game machines.
Well, maybe they've been handing these things out so frequently they've covered everything. I don't know.
No.
Oh, but now it makes sense that those were the Ig Nobel. This week, this is the real hard science contributions type stuff. So the Nobel Prizes, people who are within the Nobel Academy and all, there are a lot of nominations. People nominate different individuals for science they've done, for publications. There are then a group of people who look over all these nominations and they vote, and then there's a decision that is made. Now, we've talked on this show year after year after year about the lack of women receiving various awards, and this year I'm very excited. There's didn't they handle that one year already? They gave some women an award, and then we don't have to talk about it ever again. They can go back to the way it was. No, but it's very exciting. Today announced the Nobel Prize in Physics was awarded to Pierre Agostini, Ferenc Krauss, and Anne L'Huillier for their experimental methods that generate attosecond pulses of light for the study of electron dynamics in matter. And so the excitement of this is that attoseconds, if you think about how many attoseconds are in a second, I don't even know. I have no idea. It's like billions of billions. It's my goodness. Yeah, that's one of those numbers that when I do think about it, I'm like, It's a placeholder for something very small.
It's very, very
Don't-
small, but it is the time frame at which electrons tend to do stuff. So, in terms of being able to visualize and resolve molecular interactions that are happening at the subatomic level, the movement of electrons, the interactions of electrons, being able to create these incredibly short, like me saying short is longer by billions and billions of times than the attoseconds at which these pulses of light have been generated. So they're getting down to the limit of a photon, and also the limit of what we can visualize and what we can actually work with. So it's a very exciting. I mean, what they've done is exciting and deserving of this kind of award, because it's going to impact communications. It's going to impact our scientific investigations at the subatomic level. It's going to allow us to do all sorts of things. So, yeah, pretty cool. And that's like, go ahead.
Oh, well, I just don't understand what they did. I would love to.
Yes. So that, you know, because you could turn a flashlight on and off, right?
Yeah.
And it's like, when you turn it on and turn it off, that's a pulse of light.
Yeah.
But they...
Yeah, but that pulse of light is like billions and billions of electrons.
But billions and billions of photons and...
Photons, which are packs of electrons.
I mean, if you think
so it's even more...
If you think of how fast you can turn a flashlight on and off, it's maybe the fastest that a person could do it is like on off, like half a second, you know, maybe. But this is billions and billions times smaller than that, faster than that. And so they're using technology to control lasers to go on and off so fast that because the light is such a short wavelength and such a short amount of time, it's like a piece of film. So like when we watch movies, 24 frames per second, each one of those allows us to be able to visualize. This is like getting at the speed of the things that are working at this subatomic level. So we're actually approaching the speed of the stuff that's going on much faster than we are. And so it's allowing us to get to that point of maybe, you know, filming it, visualizing it, being able to capture it,
but
understand it, have light interact with a photon, and suddenly you see where it is. How did that photon get scattered? How did the electron get energized? I don't know how they did it. I mean, I'm not.
Well, it's not that part. It's like I still don't understand how you see anything with it, because I still will never understand how you can try to look at an electron with another electron. Like, it just
no. But you're not looking at an electron with another electron. They're using laser light, so it's pulses of photons.
Which photons, which is I think of then as just like a whole bunch of electrons.
Really fast moving photons, yeah, but. Those photons hit electrons, and then they can transfer energy or get bounced off. And the photons that get bounced off the electrons have a different energy that we can then detect. And so that gives us an idea of what happened in the interaction.
Okay. Well, I'm glad somebody got a Nobel Prize for that finally, because...
But yes, there's a lot of very interesting stuff going on there.
David Sabine Hoffender covered it. Has anybody else seen her show? It's very good. It's a very good show. I like Sabine very much. I'll have to go check that out later. But yeah, she's fine.
I have my own thoughts that I'll keep to myself. Anyway, moving on to the Nobel in physiology or medicine. Kitty, Katalin Kariko, and Drew Weissman got the Nobel for their discoveries concerning nucleoside base modifications that enabled the development of effective mRNA vaccines against COVID-19. And so this is fantastic because they have really recognized the people who did the work to begin what enabled us to have the vaccines that we have now for COVID-19 and that are going to be cancer vaccines and that are being used in all sorts of different applications at this point in time.
I should have done my homework for the show. I think one. What was the lady's name there? Who just got this? I just
Katalin Kariko, but her nickname's Kitty, which I like. But she has a wonderful story, and I don't know if
she's
it's just
the one that was like denied tenure.
Yes, and she slept in her lab, and she's had but she's also just a giving person, and now she's like, I want to mentor people, and I'm in this place now where I can give back, and everything I hear about her now, I'm just like, oh.
But she should be the poster child, for sort of what's wrong in academia. The fact that she was denied tenureship as a professor and her work was not taken seriously enough
for so long, and then ends up like maybe saving millions and millions and millions of lives. Mm-hmm. I mean, she's the one who kept pushing it forward. It didn't have to keep going. No. People didn't have to keep working. She pushed it forward, even though it was she knew there was something there.
Yeah, and this was decades ago.
That's decades. And
so we could have gotten to the pandemic and be like, no worries. We got this. No. It's going to take 20 years, but we're going to get there. Instead, we were ready.
Yeah.
Wow.
Nobel Prize in Chemistry this year goes to Mongi G. Bawendi, Louis E. Bruce, and Alexei E. Ekimov for the discovery and synthesis of quantum dots. Quantum dots are these nanoscale physical structures that react with light in a quantum manner, so that their structure actually allows us to have OLED screens. It allows us to have a whole bunch of sensors that we wouldn't normally be able to have. The quantum dots are a fascinating development, and this is a deserving technology for chemistry.
Wait, they made flat screen TVs possible?
They didn't make flat screen TVs, but this is quantum phenomena, and these little particles, they're semiconductors, but they're really, really essential for LEDs, these quantum dots. They're also being used now for mapping biological tissue because of the way they can be used to bind to different substances and molecules and the way that they interact. The Royal Swedish Academy of Sciences said, in terms of size, it has the same relationship to a football as a football has to the size of Earth. So these are these little teeny tiny dots.
That's quite different.
Like when you think of a pixel, go smaller.
Ooh.
Yeah. But they, in the 80s, Alexei Ekimov achieved the size-dependent quantum effects in colored glass via copper chloride nanoparticles. Louis Bruce later proved in fluid-suspended particles that they could do it. And then in 1993, Mongi Bawendi made it practical for production and use. So all three of them are sharing the Nobel for quantum dot technology. And who knows where they will go in the future. Again, I think a lot of these Nobel Prizes, the reason that they're exciting is that they're not just one-offs, and it's not just, it's a discovery, but it is something upon which future science and technology is going to be developed, and it's going to lead to new things. And I think that is it's a door-opening discovery, not an end in itself.
Well, they're doing it sounds like a little bit more applied.
Yes.
Waiting until something has
Now.
gotten a good application.
Yeah, it seems like they wait for that now, which is
Yeah.
probably why a lot of people die before that they get the Nobel Prize, because sometimes it takes a long time for technologies to actually or discoveries to actually be applicable.
Yeah. So, for those of you out there still waiting for your Nobel Prize in string theory, chances are you will never get the good parking spot on campus.
Keep waiting.
By the time, if you do get it, and you're still alive, you probably won't be allowed to drive anywhere.
Oh, dear. All right. Well, those are the Nobel Prizes so far. There will be more in literature, and there are many more Nobels that are given out for peace and other categories. But these are the big science ones. Mm-hmm. And so I think they're always worth a shout out. Let's talk about Stonehenge, or like, I don't know, Stack of Rocks Henge. Why?
Oh, the headline that caught my eye here, though, is a Stonehenge study upends a 100-year-old theory, suggests further discoveries to come.
Okay.
This is a team of researchers at the Department of Geography and Earth Sciences at Aberystwyth University, UK. Might be how you pronounce it. Might not be. They've made a discovery surrounding Stonehenge Stone 80, also known as the Altar Stone, and suggests it did not come from the same source as the other stones. So there's kind of basically two sets of stones that make up Stonehenge. There's the really, really big ones, which are really, really big. Some of them weigh as much as 55 tons. And those ones came from 15 miles away. They've identified where those rocks were sourced. They're sourced somewhat locally, but 15 miles is still... That's
a distance to drag 50-ton rocks.
And because this is 5,000 year ago man and women who probably didn't have a whole lot else to do, and so they decided we would move big rocks from one location to another. Because you gotta do something, you know. And weekends here, what are we gonna do? Let's move some big rocks around. And then it became a thing. Like, one person started to do it, and then other people were like, oh, I'm gonna also help you move that big rock because I'm also bored. So that's where the big ones come from. Now there's all these smaller stones that are laying on the ground and around, and these ones they're called the blue stones, and they're called blue stones because if you get them wet, they have a little bluish hue or tinge to them. And those rocks don't come from the local quarries. Those rocks have been identified as coming from about 140 miles away.
But they're smaller, so it would be
much
easier
smaller,
to
but
drag.
they're still big rocks. They're still really big rocks, but they're not 55 tons. But some of them, would still require 140 miles of many people dragging them, or being carried on the backs of mastodons, as I picture it. But
glaciers? Did glaciers do it?
No, so these rocks are actually, they're from the Redstone Quarry. Okay. Not quarry, it's not a quarry. Redstone, I'm going to mess it up if I don't look it up. Redstone Formation. The Old Red Sandstone Formation. That was created 400 million years ago when Europe and North America collided. So that stone formation is actually on both sides of the Atlantic, up into Greenland, up into Norway. And that's where they can tell that these came from. And the closest source of that is 140 miles away from where Stonehenge is located. So it's a very specific type of rock that's being used. And they have, past many studies have looked at these rocks and said, oh, yes, they came from this region, this Welsh basin, far, far away. Okay. Except the altar stone. The altar stone's composition is a little different. The barium levels are wrong. The composition of the mineralogy involved is off. It doesn't look as though this altar stone, which I think is the largest of the blue stones, doesn't look like it came from the same location. And what it suggests is it may be a much younger formation than the 400 million year old red sandstone formation. And they think it could come from as far away as maybe Scotland. But they don't know. It's a mystery now. And so now they're going in search of the altar stone origin of Stonehenge. Now,
but why? Why bring all these stones together?
Okay.
What?
I have the answer. So there's all those astronomical theories. It's like, oh, because it's a clock looking at the stars, which very possible because, in really ancient-y times, that was the only thing on the big screen was the stars. But this is also England when you probably couldn't see the stars very much because it's always foggy, right? Well, out of the year. So what do you got? And they've gone back and tested a lot of these astrological theories and found, they can fit for a minute, but they're not really great. And so those have mostly been debunked. The real uses of Stonehenge that are archaeologically discovered, that have evidence behind them, have all been found before, were all found after the thing was built. So it was built over, many hundreds of years. It started 5,000-ish years ago. And they say, okay, so it initially was also a place where they were burying the dead. But when you say initially is about 500 years after they think it was built. So it would sort of be like if modern humans went to a medieval castle and started burying people there, and then future later humans were like, oh, the purpose of building the castles is so that 20th century man could bury people there. Like, it's even the whole earliest evidence they have seems somewhat disconnected.
It seems it's all a little suspect.
Yeah.
Yeah.
So they don't quite understand how that works. Now, the point though is it's a place. It's been used as a place of sacred healing. Mm-hmm. It's been used as a place for contemplation. If
selfies.
There has any sort of exactly selfies is the current modern use. Yeah. We do a lot of selfies there now. That's the ritual that the current modern human is using. But if there was a local tradition, Druidic, town hall meeting, PTA meeting, whatever was going on, if you lived near Stonehenge,
right, come gather here.
You would incorporate
we'll all go.
The henge into whatever your thing was, because it was just too cool to not use. You're gonna have your town meeting in the musty, moldy smelling little convention hall that you built the old soggy wood.
It's not even a convention hall.
Take everybody out. We're going to the henge. Yeah, we're gonna talk about the town's business, and then we're gonna have a party.
The beginning of festivals. Yeah.
Yeah, and so it's great, perfect. That may have been actually the best explanation. You build something like this, it becomes a gathering spot. And so maybe that was the whole point in ancient times. They're trying to, they had this 5,000-year-old Neolithic village. They're like, hey, how come nobody ever visits? Oh, what's here? Oh, I know. We'll create a tourist attraction, something for people to come from far and away and trade their pelts with us. It was the first tourist attraction.
But I like also the idea that, you know, the different stones had different colors when they got wet. So this is Wales, this is, you know, a wet, rainy area. This is, you know, probably misty and damp all the time, or maybe you'd get it wet on purpose during like the harvest season or something, like depending on what time of year it was, maybe. But it's interesting that different aspects of Stonehenge incorporated stones that had different coloration and different components. And so the altar stone was different. It's this red stone versus the blue stone versus, you know, and so it's decoration. It makes it fancy. They didn't have LED splash lights back then. They had maybe fire.
Yeah.
Right?
Well, they would have definitely had fire.
Exactly.
Yeah, very much.
Yeah. But that's cool. That's really neat.
And back pain.
And back
because those rocks are really big. From
the very, very heavy rocks that they were sending, for sure. Okay.
You know what's interesting though, and sorry. Last thing I'll mention about Stonehenge, and I'll never mention it ever again.
Ever.
Never. I will never talk about Stonehenge after
Liar.
this. No, I'm not. No, I'm done.
Okay. Yeah.
But you know, like how everybody takes selfies nowadays, like you were pointing out, like it's very common. Where's the ancient Stonehenge art? Like, where's the Neolithic cave painting from 200 miles away, where somebody has got a cave painting of them, and the Stonehenge is like in the background? Like, I went there and saw that. Like, where's did that not get invented yet? Like,
it should have been. It would have been 5,000 years ago.
It seems like it would be an impressive thing, or even not even 5,000 years ago. Like the medieval Britons, the Dark Age. Yeah. No, I guess it would be. Yeah, is that the no?
No,
when is the
5,000 years ago.
Older than that.
Like,
older than
that.
Medieval.
Yeah, 5,000 years.
Oh no, I know the 5,000 years is older. When's that Dark Age? Is that post-Roman? I guess it's before the Middle Ages. I don't know when stuff happened before medieval, but post-Roman was like the Dark Ages, and like the Roman things that were built throughout London decayed, and the people were left uneducated. They didn't know who made them.
Right.
Like the memory of British history was just completely lost on the people living there. Yeah. Looking at this old ancient Roman ruins from a century or two past and having no idea who built them. Like there's an insane disconnect historically between the Roman occupation and then this sort of period of gosh knows what was going on. But there was but Stonehenge fell into that. Yeah. Stonehenge became somewhat inseparable from Roman architecture in terms of when it was built. So that there were people that thought that this
even though it was
built,
older.
Yeah, because the people didn't know.
Yeah.
And so the whole Druidic mythology that we have about Stonehenge came about from people thinking it was built after the Romans.
Right.
That this was a Druidic culture that had built it post-Roman. And so there's all like almost everything. Like if you ever see a headline that says, mystery of Stonehenge, it's just usually meaning misinformation about Stonehenge.
Yeah, there we go.
Finally debunked.
The misinformation. Again. Yes, again. Yeah, finally we're identifying it and debunking it. Yes.
Because almost everything anybody's ever thought about Stonehenge has been wrong. And that's why I think it was probably just a tourist attraction, something to do on the weekends, because it actually is a better explanation than, or as good as, anything else that's been put forward.
Or, pre-Roman agricultural festivals, harvest festivals, things related to the solstice, like all sorts of weird, I don't know, whatever. Could have been an agricultural calendar.
I don't think so. You know what, farmers?
Why would you bring the big old stones for?
Well, here's the problem with the agricultural calendar thing. Farming agriculture societies had work to do, starting early in the morning till the sun went down. They didn't have time for big rock moving. Now, if you're going up and down the countryside and you're hunting, then you have time to we'll go get the rock and then we'll hunt our way back and we'll hunt our way there, whatever. You can hunt and gather while you're rock collecting, but you can't if you're a farmer. You got to stick where you are and do your farming work. And they could tell the seasons by the seasons and the stars and the leaves. They didn't need
very square rocks. As opposed to like something that would, I'm a farmer and I dug these out of the ground. So anyway, we're going to move forward past Stonehenge because we want to do a tight 90 here.
I will talk faster from here on out.
Okay, so a study published in Molecular Biology of the Cell. Researchers at ETH Zurich have identified what they call the exclusome. The exclusome. Anyway, it's like plasmids, which are rings within bacteria. They're rings in cells. These little plasmids they have little membranes. But these are DNA rings. And they're not in the nucleus. They're not chromosomes. They are in the cell body. In just the mushy stuff that's outside of the nucleus. And so the question is, why is this DNA outside of the nucleus? What's going on there? It's not necessarily DNA that is from a virus, but it could be. It's also potentially, they think, DNA that the nucleus decided isn't necessary anymore and kicked out. But what they did is they looked in their study that they've just published at these exclusomes, and they've shown that the cell nucleus weeds out these DNA rings and deposits them with these plasmids in the cell plasma. And they have a membrane around them, kind of like the nucleus has a membrane around it, except the nucleus has a membrane that as it develops, it fills in. Like the gaps get all filled in, and it's a solid structure. The exclusome has little gaps, and it's like a baby nucleus. Like it tried, but then it couldn't, and it didn't. And so what they're trying to figure out is whether or not this is like a hygiene function of the cells to protect chromosomes. These plasmids were kicked out of the nucleus. Maybe they have genes from viruses or bacteria that the nucleus doesn't want in there. Maybe they have the potential to disrupt cell physiology. The big question, though, they also have in certain cancer cells, they have telomeres involved. And so there's a huge question now about whether these little rings of DNA, whether they are involved in immune responses and whether the cells actually, like in lupus or multiple sclerosis or other autoimmune diseases, whether there's something happening that that the cell reacts to the DNA that's been kicked out of the nucleus. It's not supposed to do anything anymore, but there it is floating around in the cell. And then the immune system responds to it. So they know that there's protein that binds onto the DNA in these rings. The proteins that bind onto them can signal cascades to release inflammatory substances within the cell. So this might, like if these rings got in there, it could tell the cell that there's a pathogen, like a virus or something that needs to be kicked out, or maybe the cell needs to die and then maybe apoptosis needs to happen to control it. But with these exclusome rings, they can't really get in there and attack it and kill it. And so it's a continued response. And the researchers say the body keeps getting the signal that the problem is still there. And so the pro-inflammatory signaling cascade doesn't subside, but continues and continues and continues. But we don't really understand them. And now this is just a new thing that we're finding, that there's this structure that's like an organelle, that's something in there in the cell, and it's impacting the functioning of the cell. It's impacting the immune system and how everything's functioning. And they're wondering if maybe this is a callback to early cell evolution, that maybe the exclusome was like the early attempt at making a cell nucleus. And so our cells kind of still let it happen, even though it's not necessary anymore.
Yeah. So these are healthy cells. This isn't connected to any pathology.
No. Nope. They're just there in our cells.
And they don't know that they contain anything. They don't know that they're holding on to anything.
They're just DNA rings.
Because there was...
Yeah.
So, two different things. One is there's an organelle that was recently discovered that was sometimes hoarding phosphate. Would that be right?
Mm-hmm.
Something like that. Okay.
Right.
And was only releasing it in times of stress.
Yeah.
When the body wasn't producing its own. So it was sort of like a reserve. So there's still hidden mechanisms in cells. You would think of cell, because you got the picture of the cell in the textbook, you think we got it all figured out.
We got it. Yeah.
But there's other little things. So I would love to see this thing studied for the other thing that is interesting is I was just reading about a study on Alzheimer's. I want to say it's Alzheimer's.
Right.
That was being caused by mitochondria kicking out DNA that was then leaving the cell,
right,
and then could go to neighboring cells, and it was creating this cascade effect of all the cells trying to kick out this unwanted DNA portion.
Yeah.
And that was creating like a. So it'll be very interesting as they track this and other, like, my goodness, here we are, the height of our biological knowledge, and we're still discovering stuff at the cell level.
Right. Well, I mean, we're just in the last decade at the point where we can actually start really looking at stuff at high resolution. There's so much happening, especially in terms of not just slices of the cell or just, it's a snapshot, but like the cell and films of it and how it works. And yeah, there's so much we still have to figure out. And of course,
it's too bad we don't have like a super at a second fast pulsing
lasers. Oh my goodness.
See this stuff.
That's right.
In real time, we could understand
we've got
more.
Cool fast x-rays and all sorts of stuff. All right, what do you have next?
Do I have something next? Oh, it's time for Justin's Animal Corner with Justin, the new normal segment of the show. That's completely new.
It's completely new. Okay, just in time.
The chilling tale of snowflies, self-amputation, survival, and certain death. So this is a story about snowflies. This is published in Current Biology. And the research details how the snowflies, which can survive and still be moving around in sub-zero Celsius temperatures, below freezing temperatures, as far as they've even recorded as low as -10 below freezing.
Which is unusual for flies.
Which is unusual for any insect. Most insects are just dead at this point.
Which is why we like to live in cold places, so that the parasites don't kill us.
Yeah. So these snowflies, they can be seen at high altitudes in the Pacific Northwest, living in the glaciers and the mountains of Washington and California, maybe as well, when there's snowpack. But they skitter around these little what are they called again? I gotta find the what kind of insect is it? It's not just a fly. It's
You're the one who knows these things.
I'm the one who knew it a second ago. Crane fly. Thank you, brain.
Hey,
Flightless.
Brian,
They're flightless crane flies. Flightless is the important part to point out because they skitter about on the surface of the snow. It's thought that they lay their eggs below the snowpack, and where it keeps a nice steadier temperature, sort of insulated. There's been some question about how they survive in these cold temperatures. And they they enlisted a bunch of citizen scientists, skiers and mountaineers, people who are going up where the snowpack is. And they said, well, if you see any of these, catch them, bring them back, and there will be a bounty. Big reward. You'll be contributing to science. And they collected about 256 adult snowflies, 20% of which were missing one or more of their legs. So they got them back to the lab, and they used some thermal imaging to observe how the snowflies maintain their ability to walk, even when their average body temperature is well below freezing. And at some point, ice crystallization begins to form within the fluids of the leg. And this spreads pretty rapidly. It takes about a half second for that frozen leg crystallization to move up the leg and into the body's vital organs. But within that half a second, the snow crane flies do something really interesting. They just kick off the leg. Boop! The leg is gone. No more crystallization, no more icy death. And what they found was that this is actually something that that exists in other crane flies and actually a number of other insects, that is usually triggered by something grabbing hold of and pulling on the leg. So it's sort of like lizards that have the detachable tail. Catch a lizard by the tail and they just let you have it and take off.
I've never actually done that. Have you done that?
No, I've never done it.
I've never actually done that.
Actually, have I? No, I don't think I have. So snowflies. These snowflies have, as soon as their leg begins to freeze, they just get rid of that leg. Now, in terms of a survival condition, it may not seem like getting rid of one of your legs is a great survival, but they've got others. And
they've got 6. We have 2. It's
crane fly life isn't that long anyway. Like, it's better to live with 5 legs or 4 than it is to die today. It's one of those. But the thing must happen like automatically, because they also notice that when you pull on a snowfly's leg, unlike other crane flies that have these sort of detachable legs, they don't come loose. It's not a mechanical trigger anymore. They think that there are neurons in the leg that are monitoring temperature change, and so when that crystallization event starts to take effect, they get rid of the leg. They kick it off.
I mean, you don't want frostbite to result in gangrene that then results in sepsis that then takes you out. So if you can naturally avoid the process that is going to take out the whole organism, it's going to be better overall.
And in this case, it's that ice crystallization that takes place in about a half a second. reaching the vital organs. So it's got to be pretty much automatic and quick. You got to be ready. You always be ready to self-amputate. It seems like a kind of an extreme environment to choose to live in. Like, aren't there warmer places that the snowfly could live?
But it likes it there. That's where it lives.
It likes it there because there's also not very many things that eat crane flies at that temperature. There's not a lot of predators around.
Right, but what do they eat? So what's the ecosystem for these flightless crane flies? What are they eating? What do they do?
I don't know what they're eating, but I know this is because it lacks actually they eat nothing.
Nothing.
They only drink water. So
That's
they their larvae or whatever, like do all the eating, and then once you're a full-grown crane fly, you drink water, but you're done eating. So they don't even need the food.
Right, they're just there to breed after they're a larva.
Just there to breed.
Right. Okay.
And they breed on the surface of the snow in full view. They have been observed mating for 30 minutes or more. This is an unusual activity, because normally if you did that, any other crane fly would have, for 30 minutes exposed like that, would be eaten. So they're very well adapted, and they're enjoying all the benefits of not having any predators. They lay their eggs beneath the snow. That place is not really disturbed. There's nothing, again, they're hidden away. It's such a perfect environment for them that it's worth amputating a few legs once in a while to have such a nice environment. Problem is, it's going to probably go away by
climate change, all that kind of stuff.
Yeah, the snowpack where the snowflies live is predicted to be gone. 70% of it will be gone by the 2080s. Loss of snowpacks predicted to increase thermal variability and decrease temperatures under the snow where the snowflies lay their eggs. Also, the things that they eat, the plants likely won't be surviving for a whole lot longer. The freeze-thaw cycles.
I'd love to know, though. I mean, this is a whole thing as glaciers shrink and the ecosystems change. So we know mosquitoes are food for lots of organisms.
Yes.
These snowflies are they food for a lot? Like, what are they if they are unable to have the ice to lay their eggs on or the water? What is that going to impact downstream? So, like, you have to think of it in the food web perspective. How is the ecosystem going to be impacted?
Yeah, so you know how I was just saying, like, they don't have many predators because of the temperature?
Yeah, but they have some.
The predators that do rely on them also don't have a lot of other options, right? Anything that's eating a snow crane fly,
there's not much else to eat.
Doesn't have a lot of options. So that impact in an environment like that is going to accelerate quicker, be more noticeable, move up that food chain a lot faster than it might in otherwise. A more diverse, abundant life environment. So.
Interesting. Okay. Now I'm depressed, but let's not talk about
let's talk about something less depressive.
Let's not. I have cats. Cats are nice. I know you don't approve of cats most of the time, but let's talk about cats. Cats purr. Their purring is awesome.
You know what that sounds like? What are
you going to say, death?
Very suppressed aggression.
No, I've got concealer on today, actually, because the other morning my cat was sleeping with me under the blankets and got scared and clawed up my whole body and face to get out from under the blankets, and I have scratches across my whole
Oh
forehead.
no.
Which is pretty funny.
Oh no.
I was like, that would have been a nice morning to lose my eye. Okay, great. Anyway, that didn't happen. I love my little tiny predators that I have in my house, that I've kept inside, who may or may not have toxo. I don't know, but I love them, as crazy as they are. And then they come and they cuddle and they go. But see, I make that sound and I have to breathe in, and I can't keep making that sound because I'm making it with my tongue as opposed to my vocal cords. And it turns out, so cats produce the purr with their vocal cords. And there has been a debate about how cats produce the purr for a very long time. And so in this study, researchers were like, well, we'll just take cat vocal cords, put them in dishes, and see what we can do with them and see what happens. Turns out, cat vocal cords are really awesome. And they're kind of similar to the big cats that roar. Their larynxes produce the purr without necessarily having to have that like inhale, exhale, nervous connection. So that it's like walking, where once you start walking, you keep walking until there is a command to stop walking, So there doesn't necessarily have to be nervous stimulation to keep the purr going. The purr just happens. And so these vocal cords are also specially structured, and they've got this, what they call a pad on them. Our vocal cords don't have pads. We have singers who like to sing really loud and pretend that they can do like vocal fry type stuff, and then they get polyps on their vocal cords that have to be removed, and maybe they won't be able to sing anymore. Well, cats, they have these pads that are like, we're already there. We're padded. We have insulation. Everything's great. It's going to be good. It's all okay. They did these stimulations to cat vocal cords and found that this pad is very special, and also that the innervation of the vocal cords, the trachea, the larynx, it's all very unique. And the cat purr it's a very special thing that the way that they've kind of like Stonehenge, breaking old ideas. This is like a study because they've looked at the anatomy and the physiology and the neurophysiology. They've turned over old hypotheses of how purring happens. And we have a new understanding of how cats purr.
At last.
At last.
Long last.
At last we know why and how the cats can purr. It's very exciting. And so now the question is, do we need a new theory of purring? Maybe we do. But anyway, these vocal cords make very low frequency sounds, similar to human vocal fry, but a little bit lower. And they don't consciously control it.
That must be annoying. What? Right. Is that my fridge? No, that's you. You're doing it again. What am I doing?
I love this. In science, they have a quote from Bonnie Beaver, a veterinary scientist at Texas A&M University, who was not involved in the study, but I love this quote. Purring has historically had a complex, non-scientific explanation. So we've got science to study purring right now. It's very exciting. Purring, it's like vocal fry, but better. And then my final cat story isn't really cats necessarily, but related to all sorts of mammals. And if Blair were here, she'd really, really, really love this story because we've talked about fluorescing animals a lot. This isn't bioluminescent animals, so this isn't like animals going, I'm producing light. These are animals
fireflies or deep sea whatever creatures.
No, these are animals you want to take with you to the next blacklight rave that you go to. So there have been studies where they've reported different animals glowing under ultraviolet light. And it's like, well, who glows under ultraviolet light? And so a group of researchers took it upon themselves to go through their local museum mammalogy section and expose all sorts of animals to ultraviolet light. And it turns out so many mammals fluoresce. We've got, I mean, you go under a black light and your teeth and your eyes, the whites of your eyes and the whites of your teeth are going to glow. But what
and
find...
that one trench coat I have is gonna, it lights up because it's got little, what do you call it, lint or fluel or a little
the lint on your black sweatshirt.
All of it lights up.
It does. So they photographed preserved and frozen platypus specimens because, I mean, if you're going to look and see who fluoresces, you want to know whether or not it's a platypus because they do everything else. So yes, the platypus fluoresces. It's very exciting. It's very pretty. UV light makes it very lovely. And then they looked at a whole bunch of other organisms like polar bears and wombats and armadillos and cats and bats and rats and zebras and all sorts of things. And they found that very, very, very many animals do fluoresce. The majority of them are nocturnal. So there could be something to do with maybe the transition, sunset or sunrise, kind of, when ultraviolet light is very dominant. There could be something there that's related to identification and communication. But at this point, there is no evidence that fluorescence has anything to do with anything. And it's possibly just a factor related to just proteins involved in keratin and the structures for skin and hair and teeth and stuff.
Okay, but hang on. Wait a second. But didn't you just give us the connection that it tended to be nocturnal animals? Or is that just...
No, it tends to be.
Or is that just what they happen to look at? Because that sounds like a connection that seems like it would be very important and not just a coincidence. And I would now also want to know about the vision of all of these animals to see if they have some infrared vision and if that became a selection for mating because it would make them easier to find each other over time and things like this.
There you go. Yeah, we don't know. So these are the next questions, right? These are the directions that this research will now go in because it's like, hey, why? Oh, they're fluorescent. What's going on? We just looked at mouse pee. And we know that fluoresces, but geez, there's all this other stuff. So it's a big question as to why. Why do nocturnal animals seem to fluoresce more than others? Aquatic species are less fluorescent. Burrowing animals fluoresce a lot also. So, what's happening there? Is it just that hair is heavily pigmented or is there something else going on? We don't know. But
if they can figure out how a cat purrs, they can figure out pretty much anything.
Yeah, and so
this has been Kiki's Animal Corner. You see how we did that? It was Justin on the way in and Kiki's on the way out.
We pulled it all the way
Gary
around.
is asking, is Blair ever coming back?
I don't know.
I sent her on assignment to the Stonehenge.
She's doing children animal studies right now.
To investigate the henge. And then the study came out that it was like overturned the thing. Anyway, so it's kind of ended up being pointless, the trip. But she's still there. She has not come back from the henge. And we're still awaiting Blair's return from
Childhenge. And anyway, when I told my family about animal cat fluorescence, of course, my husband pulled out a black light and they grabbed the cat.
Because of stop.
Yeah,
just pause it for a second. Hang on, pause the story.
They forced my cat into a rave.
Pause the story for a second.
And she fluoresced.
Okay. Your husband, hang on. I said to pause the story. You can tell me whether or not the cat fluoresced. But here's the thing.
Yeah, I know.
Your husband
has black lights.
A grown adult man.
Yep.
And you're like, oh, yeah, there's a fluorescent story. He's like, let me go get my black lights.
Which he bought at Home Depot for like $8 and he's super excited about because they're like LED black lights and they're super like energy
so they're replacing
efficient.
His old black lights. Okay. I just want to be clear.
Oh no! He's like, look at this! It's a really cool light, and look at how they built it, and blah blah blah. He was very excited about his black lights.
Don't get me wrong. I'm completely jealous of the fact that I wouldn't have even thought to look for black lights in the last 40 years, 30 years, but 20 years, 10 years.
Yeah.
But
yeah.
Anyway. Yeah. And so your cat did fluoresce.
Yeah, he said, "I'll be right back," and ran outside, grabbed the black light, came inside, and they wrestled the cat.
It's also knew where they were. Like I could understand, we have black lights. They're in the attic, and they're in a box, in one of the boxes in the garage. He's like, "Nope, got this. Know exactly where I've got labeled on the shelf. Here's the black light in case of emergency. Got a mobile battery pack so I can carry it around."
Yeah. Now my cat's sitting here on the couch, looking very cranky. Sorry.
I'm sorry. I because I kept pausing everything. I missed everything.
But she fluoresced.
She fluoresced.
She was fluorescent. She has a lot of white fur. So yeah.
Did you compare it to your other? You got like 6 or 7 cats, right?
No. I have 2 cats, but nobody else but me can pick up the other cat, and I wasn't in the room, so we're gonna have to do the experiment over again.
All right.
I will report back.
But I also want to just to be sure, let's get some non-nocturnal animals. I don't even know what that is. Squirrel?
Oh yeah, we have all sorts of
Squirrel nocturnal? I don't even know what's nocturnal anymore. It seems like
I don't know. I would love to trap the squirrels and the bunnies in my backyard anyway, so I could do that.
Oh, bunnies probably aren't nocturnal.
The bunnies keep eating my plants. Anyway. This is This Week in Science. Thank you so much for joining us for this exciting adventure into cats, Stonehenge, purring, fluorescence, and so many other things. We love that you are here joining us for this episode. Thank you so much for joining us. If you are enjoying the show, please head over to TWIS.org and hit the Patreon link. Patreon is really how we support ourselves. We're listener supported. So you are a huge part of supporting what we do each and every week. Without you, we really can't keep it all up. So I implore you to head over to Patreon, to TWIS.org, click the Patreon link, and $10 and more per month, and we will thank you by name at the end of the show. $15 and more, you get stickers, and I think at the $20, $25 level, there's a t-shirt involved. There's a whole, different levels have different things. But whatever you can afford, really, any amount helps. Honestly, everything helps keep this show going, and we depend on you. So thank you for being here. If you can't afford to help in the financial sense, just share TWIS on social media. Share it with your friends. When you're talking with someone at a cafe, go, "Oh my gosh, I heard the story on this podcast called This Week in Science. Why don't you subscribe right now?" Get them to subscribe. That would be an amazing thing also. And I have another cat back here. Hold on. I don't think you can see her. Hi, what are you doing? I don't know if you can see her. Nope, she's below your eye level. You can see her ears. You can see her ears. She's below eye level on the shelves. But anyway, TWIS.org. We also have merchandise, and if you are interested in TWIS merchandise, we have hats and mugs and bags and towels and shower curtains and all sorts of things, really. If you go to TWIS.org, click on the Zazzle link, you'll be able to peruse our store and see what we have to offer. The holidays are coming up. So for those of you who do know people who enjoy TWIS, or even if you want to buy yourself a present, Zazzle's where it's at. Oh my goodness. We are still going with more science and sanity. And yeah, I don't know when Blair's coming back. We haven't talked to her lately, but I'm sure she will be back very soon. Let's talk about mushroom microdosing. How does that sound? How many of you out there are in a place where mushrooms are relatively legal, or at least not completely illegal? It's interesting here in Oregon, and it's been a weird little bit of time. Psilocybin has been put forward similar to ketamine and other substances to help with depression, to help with anxiety, to help with a whole bunch of psychological issues. And there's a study out this last week. Hey, Justin, you're back. There's a study out this last week in Nature where it's Molecular Psychiatry, one of the Nature journals. They gave psilocybin to rats, but not big doses, microdoses. So the idea of a microdose is that it is a sub-hallucinogenic dose. It is a like the dose that would normally make you feel the effects of the psilocybin. It's lower than that. And so there is an idea in the psychedelic community that LSD, mushroom, psilocybin, microdosing can be beneficial to behavior. And that it can be beneficial to, instead of taking citalopram, instead of taking Zoloft, instead of taking any number of antidepressants, that these microdoses can be helpful for a number of things. There have been studies with people that have been kind of equivocal as to what's actually happening. Some studies have said there's an effect. Some studies have said there's no effect. Like there have been many studies where it's like, hey, people took microdoses of LSD or psilocybin, nothing. There was no difference between the control group and other groups. When we're talking about...
and then to clarify, there's other studies where they did macrodosing.
Yeah.
Where they saw really positive results. Yeah.
And differences between control groups and not control groups.
Yeah. And positive and very positive outcomes.
Yes. And so there's an interesting question right now about, how does, if we give little teeny tiny doses over time instead of big doses at one time, how's that going to impact people? And the problem with human self-reporting or with any kind of involvement where people are answering questions about how they feel or what's happening.
The problem with the field of psychology is...
Yeah, the self-reporting bias is an issue. And we also understand the placebo effect is an issue. If you want to have an effect, you might think that you have an effect. And so here is a study with rats. Not humans, with rats. So it's not people, but they... gave these rats microdose-like regimen with repeated low doses of psilocybin at a dose derived from occupancy at rat brain 5-HT2A receptors in vivo. So what they're doing is they're looking at the lowest dose of psilocybin that they could give these rats that would lead to the serotonin receptors and a particular group of serotonin receptors, these 5-HT2A receptors. And there's multiple serotonin receptors in our brains. But these specific receptors, they created this system where it's like, okay, we know this microdose is linking to these. It's grabbing onto the receptors are grabbing onto them. And so there should be some kind of effect. They determined that there was tolerance. The rats were fine. There were no signs of anxiety. They didn't have any signs of what would be hallucination or anhedonia or anything in their, like, they were fine. They just seemed normal. The rats were okay. So there were no deficits in their behavior whatsoever. But over time, they started injecting the rats. Like, they're like, I'm going to grab you and inject you with a needle, which is stressful, right? And so stressful situations are normally going to lead to a comforting response. And some animals respond better than others. Some respond, with a little bit of grooming and then they're okay. Others go down the road of kind of compulsive activity, self-grooming that is kind of OCD levels. There's a lot of stuff that goes on. And so
which sounds like you're identifying then, like when I would say this type thing of this, amygdala. Amygdala response in those ones is not turning. Is it thalamus? I thought it was amygdala. Sending
for the stress, for the fear and anger, but the thalamus is like the big area that's related to these kind of like behavioral responses. It's connected to the amygdala, and it's all that the thalamus is all in there.
But it's sort of like it sounds like there's continually to relive the stress that is being signaled out again and again and again, and that's why they keep trying to groom or self-calm or self-soothe.
Self-calm, yeah, exactly. And so what they found with the rats that they gave them microdoses of psilocybin is that they had less of that. They had less self-grooming, less self-soothing, less of the proxies for human compulsive actions. And they also had that they looked at their brains and they saw that there were increased expressions of certain receptors related to serotonin. There was increase in synaptic density. And so things that would normally be related to potentially PTSD or responses related to traumatic events and anxious responses, obsessive compulsive disorders. This links in, I mean, it's rats, not people, but what they've seen in these rats links in very highly with work to suggest it could be useful to use psilocybin microdosing as a therapeutic intervention for certain behaviors in rats.
Yeah. And again, it sounds like it is all of those sound to me like affecting the amygdala's ability to continue. And there was there's been other research that sort of seems like it's on this vein, but that's why it's so good against things like PTSD, is because that is the amygdala, ancient part of the brain that tells you fight or flight. That in this human brain sometimes gets stuck on,
right,
and keeps sending the stress, the alert, the fight or flight message to the brain.
And it's like
because the conscious brain,
you're in a normal situation at a grocery store, but something loud happens, and that makes you associate with a previous traumatic experience. And instead of just calming yourself, you go into an anxiety or a stress response. And so and what part can we do to help?
Part of that also that's involved in that is that all of this really high level thinking that humans do is a part of the brain that sort of developed later than the amygdala. And the amygdala has communication to that part of our brain. But there isn't really a good line back. It's really hard even to think your way through rationally, logically, to soothe that old ancient-y part of the brain, because it's not built to listen. It's not built to listen to the primitive animal for input. It's got its own inputs from the sensors that it takes and decides what's to do. And it's designed to tell the animal how to react. So it's not listening to your conscious brain, which is part of the problem of addressing things like PTSD and anxiety disorders, is that no matter how logical and reasonable and rational you know that you're not in danger or there's no reason to be upset about something.
But if you
can't communicate, you can't turn off the part of the brain that's saying this, that's screaming this in you. Okay.
Sorry, my cat just attacked thing. She's climbing.
So the microdosing, it may work. Look, I know there's been a lot of push also to remove the hallucinogenic effects associated with, as though they were separate. I think they're connected. I think that the trip that you go on when you take these is part of that process.
It might be part of releasing. Yeah. Who knows?
Is part of that. Yeah. Removing the filter and allowing the amygdala to be somewhat, maybe even communicated to by the conscious brain. And so, look, I had therapeutic situations create a comfy cafe environment that somebody can chill in and have these experiences, make safe places for this to happen. But I think macrodosing shouldn't just be ruled out because people are like, oh, you don't want people tripping out. Because you know what? People have done this a lot of times throughout history. And the latest Black Mirror had somebody doing LSD and then immediately jumping off of a balcony.
What?
And somebody who apparently, according to the story, would have been experienced with this too. So it's just like, that is somebody said they saw that in a 60s Reader's Digest. Yeah. And that's about how common that would be. A story from the 60s in a Reader's Digest and a story from the 2020s in a science fiction-y show. Those are the accounts that we have of that taking place.
Yeah. I mean, I agree that we don't know enough, and I think we should be looking at all the options. And the fact that we have financial and political forces that are influencing what is being studied and what's not being studied. I'm excited, though, because over the last 2 decades, we have seen more research into these substances than previously. And it's like their restrictions have been relaxed so that we're actually starting to look at how these things can be used therapeutically, and it's really great. So I don't know. I think there's a lot of fear that has been fomented for a very long time.
And the best way to reduce it is for all of those afraid people to macrodose.
No,
then they won't be afraid anymore.
No. We're not recommending that. No. Nope.
We aren't. No, not as a group, not as a show. Me individually.
No, you're not even. No.
I can't even.
No, we're not. Nope. We're not recommending anything at all. We're just talking about this as curious human beings.
Okay.
Okay.
You know what I'd be curious to know?
Do you have more stories? That's what I'm curious about. Do you have more stories?
I do.
Okay, let's hear it.
All right. So here's a story that I feel like we've covered before, but it's new. This is published in Royal Society Open Science. It is Positive Impact of Sleep on Recall of Multiplication Facts. Basically what they did is they took 77 adults and they split them up and had one group learn a bunch of complex multiplication problems right before going to sleep, right before bed. And the other group had the same lesson first thing in the morning. Then they would go through a series of learning, being tested on that the next day, or being given new problems. So they were answering these online, and they'd have to recall stuff that they learned from an earlier lesson. And which group do you think learned better?
The ones that got sleep.
the ones that got sleep right after learning. So we've talked about this in a few different ways. One was an alcohol study, which is interesting. And one was a study, but the idea is, if kind of like in the past, we've seen like this pattern of, if you don't have competing information, the brain is able to lock in new knowledge better. So that, if you learn all of this complex math first thing in the morning, and then say you go on to a chemistry class or an English class, or you go to work and have a really busy day where you have lots of duties and tasks and responsibilities and things are shifting, and then you get home and there's conversations with friends and your spouse, and then you're doing some stuff, and then you go to, and then the next day you're like, hey, what were the math questions we asked you at the beginning of your busy day? You're like, I have no idea.
I wouldn't remember it.
But if you learn them before going to bed and then get quizzed the next night, that knowledge that you were acquiring stuck. And part of the reason, I think, is that there's nothing competing with it. So as the brain locks something into memories, there's only one current thing that it has to work on.
Yeah.
To encode.
We're going to talk about this in a little bit, because I actually have some studies related to memory in a minute related to this. Okay. So I'm interested in this. Yeah. Let's keep going.
Well, I mean, that's kind of it, I guess. Uh-huh. They don't know what the mechanism by which the encoding takes place. But they have they do suggest that sleep conferred the additional benefit on recall. Mm-hmm. They do point out that they have the limitation of the study that there is a lack of comparative stimuli with similar complexity of encoding that would demonstrate that this was sleep specifically related, or that it was math specifically related.
Right.
So they kind of leave that open. But the idea, though, is if you got a kid who
you're messed up, you're not going to remember any math.
If you got a kid who's struggling in math,
make sure they go to bed.
Make sure that instead of the bedtime story, put that aside for a minute, and maybe do the math lesson right before bed.
Or incorporate it into the bedtime story.
If they fall asleep during the math lesson, hey, they got to sleep. That's great. If they make it through, though, then it may lock it in better. It may be when that math class is. So also, if there's a subject that you're, and this is tricky, because if you're trying to plan when to have classes during a day, do you put that hard class that's going to require a lot of memory at the end of the day? Because in a way, if you put it at the end of the day, there's less things that compete with it before you go to sleep. But if you put it at the end of the day, you're also more tired when you come to the class.
Yeah.
So are you as good of a listener? Are you as good at paying attention? According to this study, that doesn't matter so much as having the time to encode. They also compared, they had people with different ability levels of learning these math problems. And they found that, you could, by taking account of who's just innately good at math versus those who struggled with it, didn't matter if they were part of the sleeping group where they got the questions and then got to go to sleep right away. Regardless of what group they were in, they measured up better than their peers on the same level in terms of recall. So it wasn't just some people are better at math. There were different levels within the group. And as those tracked through, they still saw improvements in the recall depending on who got to get to sleep right away versus who had to go off and have the rest of their day before they were able to really encode and lock these things in.
Interesting.
Yeah. So listening to this show before bed.
Right before you go to sleep. Yeah.
Or listening to this show while you sleep. You may encode.
Really encode it in there.
Gary Ellis, this is why I remember Johnny Carson. Yeah, it probably
This is why all the jokes from the late night shows stick in your brain.
Yeah.
Exactly.
And does anybody remember a single episode of a morning show? I don't.
No.
I remember nothing from
No.
any morning.
No, mornings are
not
bad
that
for
I've
memory.
watched them,
That's where
though.
we just want to
Also,
say that.
I don't watch morning shows. That might also.
It's time for morning shows. I don't
Yeah.
know. So let's talk about memory a little bit. I had a couple of stories that were specifically related to memory and how it all works. Sometimes your brain knows when it's got it wrong. And so, like you were talking about, like, okay, so maybe it's a program where you're given a bunch of questions related to math, and then you're given a bunch more, and then like later on you're asked to answer questions about those things that you've seen previously. This study was related to words and not math, but it was specifically monitoring behavior in the hippocampus in epilepsy patients while they were undergoing surgery. So these were awake individuals having their neurons in their hippocampus recorded for their activity. And the bottom line of this study that was just published out of the University of Pennsylvania is that the signals in the hippocampus change their frequency. Like they're either higher frequency or lower frequency. And the frequency of the signals of the hippocampus indicate whether or not you're going to answer wrong. Like you have a false memory. So, for example, you're given a bunch of words related to flowers. Rose, daisy. I don't remember any other flowers. But anyway, you have a bunch of words, and that's your list of flowers. And then they test you on it later, and you say daffodil. But daffodil wasn't part of the list that you were trained on.
Okay.
The hippocampus has a lower frequency response. Like it's like they've got these different oscillations, which there's high frequency alpha beta oscillations, and then there are lower frequency theta oscillations. And so theta oscillations are what are seen when recording and a person is about to say daffodil and answer incorrectly but relatedly. So it's basically what they're looking at are false memories.
So they can tell the difference between recall,
yes,
and a logical guess.
Yes.
Wow.
And so what they were really trying to dig into is episodic memory. Episodic memory is a memory of an episode in your life. So for example, every morning you get up, you get a cup of coffee, you have some food, maybe it's Raisin Bran or something like that. I don't know what you eat in Denmark, but or eggs or whatever. But whatever, like every day you kind of do the same thing, right?
Yeah.
But then maybe one day you do something a little different, but then somebody asks you what you did and you go back to your normal routine of, oh yeah, I ate Raisin Bran. Your hippocampus knows that you're answering incorrectly. The frequency of the neuron's responses is different. So, it's like your hippocampus knows you ate eggs, and you're it's like meh.
Is it know that you're wrong, or is it the difference? Like I was saying, like between the way the brain recalls a stored memory. Yeah. Versus the brain logically working to find an answer that would fit. Because that to me, like those are two different maybe processes that are being observed, and it can tell the difference between which one's actually taking place. Because I'd be curious to know whether or not I have recall at all, or if everything that I do is just a logical guess. I feel like mine would just be me assuming that that I drank coffee, because I don't actually remember drinking this coffee.
But you did.
I don't know what happened to it.
It's gone.
It's gone.
Right? Yeah, so
I assume I drank it, but I don't recall doing it. So am I going to be in the...
Right, your neurons know what you did. And then in the recall aspect, it is the probability difference of the association, right? So what is the problem? You say, I did not drink coffee. But, you come up with that answer, but there's a probability difference that changes the frequency of the neuronal response in the way that it is. And your hippocampus is involved in not only encoding memories, so like while you're sleeping after you've studied the math, your hippocampus, like you need sleep so that consolidation can occur, so that your hippocampus can go, boop, this memory goes over here. I need to make this association over here, and the networks get created. But then during the day when it all comes back together, the hippocampus again is kind of like the conductor, the orchestra conductor. And it's like, the oboes are flat. There's something not right here. So it's kind of like
flat.
So it's really fascinating that we have false recall. We can be convinced of things, like we've seen multiple times people on legal defense stands, like being kind of convinced that they've seen something when they really haven't, right? And then testifying to the fact that they've seen something. This is false recall. There is actual difference in the way that the neurons in our hippocampus respond during false recall, which I think is fascinating. And it can be decoded. And that is going to be interesting. So that this study could potentially lead to some really interesting work leading to understanding when people are actually recalling things falsely or when they're actually telling the truth.
So, yeah. I just have this really interesting. This is going to be very interesting because there are people who I have known in life who have photographic memories.
I know these people. I have a friend. You just go, this date, and she goes, yeah, I know exactly everything that happened on that day. And I'm like, okay.
And so people with this innate ability can look at some body of text, look at it, and then literally read it 10 minutes later.
They sleep.
So this is like I don't mean they read it and then remember it 10 minutes later. They can look at it and read it 10 minutes later. Yeah, that's what a photographic memory is.
It's like a picture. It's in their head.
That's recall.
Mm-hmm.
When I have sort of a vague impression of the words that I have read, that I can make sense out of because I got the concepts all in line and I can talk about them with some level of clarity, but I have zero picture in my head of words. None. Right. I have no picture of the words. I'm not reading anything. So I'm wondering if this test was applied to a task like that between somebody with a photographic memory and somebody without, when talking about concepts, not necessarily a memory of an event, but here's the thing to read versus later. One is going to be recall.
Yeah.
And the other one, I'm curious if it looks like a false memory in the way that it's generated, because it's not recall.
It's not recall.
It's concept reconstruction. So this is a very fascinating subject now. I want to get more follow up on this, and we gotta. Can we get one of these researchers on the? Let's get one. This would be a fun interview,
right? Let's talk about false memories. Yes,
I would love to see if we can get somebody involved in this, because this is a very interesting area to talk about.
Very interesting. Yeah. What is
because you and I
recall?
too,
What is
you and I too
conceptual?
have the different memory types,
Yeah.
have the different internal visualization types.
Yeah. It's
I
all over
can't
the internet. This week has been huge. Apple in the brain thing, the
Yeah.
aphantasia. Like,
Uh-huh.
everybody's like, oh my gosh, I thought everybody could see an apple in their head. And I'm like, you obviously have not been listening to TWIS. Jeez.
Yeah, no.
Yeah. Yeah. But then there's the question, like, is it the same thing for music? Like, I actually, like, hear music in my head. So I don't see things. I have a vague idea of things in my head, but I hear music. I hear voices.
Yeah.
I'm not kidding. I didn't just say that. Okay.
Yeah, you did.
Okay.
So-
let's move on to my other study related to
But
memory.
here's the thing. Here's the thing. I just want to clarify something. If the voices that you hear in your head
Yeah.
are your own,
Yeah.
nothing to worry about.
Yeah.
If the voices that you hear in your head
Are other people's?
may be a problem,
We're
if
not-
the voice you hear in your head 24/7 is mine,
Oh,
you're not me.
I'm going to tell you that is not what's happening in my head. So just,
Okay.
that's not happening. Another really interesting study that came out this week related to memory came out in the journal Neuron. And there's been a huge question for years and years and years about, like, what are the cells that support memory, right? We got the neurons. I've talked forever about the hippocampus. The hippocampus goes and, like, shunts things out, signals go places. We've got cells, there's a network, right? And then, like, when it gets replayed, that network gets turned on again, and ta-da, memory gets replayed in your head. But how does this all work? And in this study, researchers at New York University have shown that not neurons, but cells that wrap around capillaries in the brain. They're called pericytes. They work with neurons to help the formation of long-term memories. These pericytes influence, not parasites, but P-E-R-I sites. So
Pericytes. Okay.
pericytes. They wrap around the capillaries and they can influence blood flow. So they influence the amount of oxygenation. They influence the amount of, metabolite removal. Like they're influencing all sorts of stuff around these cells. And so these researchers are really excited because what they're really digging into is this cooperation between all the cell types that are in the brain. And, like, forever we're just like, oh, it's the neurons. The neurons are everything. And then we start, oh, it's the glia, and those are the ones that wrap around the neurons. And oh no, wow, there's this other group of cells that they have now identified as really being important in memory. They've shown previously that when pericytes are lost or malfunction, that's involved in neurodegenerative diseases like Alzheimer's and other dementias. They maintain the structural integrity of the capillaries. They are also an important part of the blood-brain barrier that we've talked about so many times. And they release a factor called insulin-like growth factor 2. This protein increases following learning in places like the hippocampus. And so these researchers were like, what happens if we mess up insulin growth factor 2 in brains? And so, of course, not humans. They messed it up in animals. But they showed that production of this IGF-2 by parasites in the hippocampus gets enhanced by learning. So when mice were subjected to memory tests and they had to associate a foot shock to learning to identify an object in a location or a particular place in a maze, that learning ended up releasing IGF-2 from the parasites in the hippocampus. That led to enhanced memory. And so the parasites wrapped around the capillaries are working in concert with the neurons in the hippocampus that are involved in taking those things and shunting them out to other places and making sure that long-term memories are stored. What?
Cool.
Yeah. So there's what they call now, there's a neurovascular unit regulating neuronal responses and functions of the blood-brain barrier. And we should probably be thinking about this related to brain injury, inflammation, neurodegenerative diseases, and we've never really looked at this before. So, boom.
Yeah.
So much we don't know. And then a new thing comes out and you're just like, what? Oh my God. Yeah.
Yeah.
This study excites me. The last study, all these memories.
I've got one more. I've got one more.
Oh my gosh.
I've got a last story.
Of course. Murder in Oxford.
Murder. This is a project that has been mapping medieval England's known murder cases. Has found that the population of Oxford students was by far the most lethally violent of any studied.
I thought academics were just supposed to do the academia and they're like, I need to publish this paper.
Yeah, school used to be real different. in medieval
it was more Hogwarts. I don't know.
No, it wasn't like. Yeah, it's kind of like Hogwarts. If everybody at Hogwarts had a sword or an axe on them, and then instead of like, I will out magic you in this test, it just went at each other.
Whatever.
Digital. So this is a digital resource they have called Medieval Murder Maps, which I think you can go and Google and find. I think that's a thing that you're allowed to look at now. These are investigations from 700-year-old coroner's inquests.
Oh my gosh.
And coroner used to have a job not only as the, to try to figure out who did what. They were sort of crime investigators as well as just looking at corpses. They did an estimate the per capita homicide rate and found that Oxford was 4 to 5 times higher than the late medieval London or York. So those are the big cities back in those days.
Oxford was more dangerous than
100
London.
times more. And it actually, according to this, it's actually the murder rate was higher than it is in current modern day cities anywhere in England.
I'm just playing around with this medieval murder map, which you can definitely go to, and we'll link to the story on our website, and you'll be able to get to it through this. But you can pick the genders of who was murdered or who was the perpetrator, and on what day of the week. So Sundays, there were really only like 21 Sunday murders. The weekend was not so busy, apparently. Oh my gosh. And it's all down King Street. Wait. Yeah. Wait, no.
That's
isn't
one
that the big
street. You're looking at the murder rate of one street.
Seriously, it's like that's the big street. Isn't it? Oh, High Street. That's like the big street in Oxford.
Yeah,
everybody would go to the pubs and then murder each other. Okay.
Yeah, that's basically what they kind of found, because this was, according to this, they said Oxford had a deadly mix of conditions. Students were all male and typically aged between 14 and 21. At a point when, young men are usually kind of risk-taking. They were all freed from being, around family or their local priest or whatever. They were put in this environment away from supervision. This environment had lots of weapons, lots of alehouses, and lots of sex workers. And there was a very diverse population at the university. which had, maybe only 1,500 students, but they were from all these different parts of England.
Mm-hmm.
And they all had very fierce sort of rivalries with each other that they brought with them, regional rivalries and this sort of thing.
Can't y'all just get along? Just study. Come on, peeps.
Yeah, so according to this, a Thursday night in 1298 saw an argument between students in Oxford High Street Tavern, resulting in mass street brawl with swords and battle axes. So also, by the way,
So I'm loading up this one here on Thursday night. It's a poor scholar accidentally smites Matilda and kills her.
Actually, yeah. So Matilda was a sex worker.
Oh, Matilda.
Who he decided accidentally didn't want to pay, and so killed her.
So I'm gonna smash your head in. Mm-hmm.
Yeah, or I think it was a stabby fella.
Oh. Thereupon Robert smote the doorkeeper 2 times with an ashen staff which he bore in his hand. When he would have smitten him a 3rd time, Matilda, who was standing by, leapt forward between them so that she received on her head the blow which Robert purposed to have given the doorkeeper.
Oh, no, that's a different story than the one I was even thinking of.
That's Matilda. Poor Matilda. Oh, there's so many. Students attack a constable of the peace. Harps, viols, insults, and a stabbing.
The researchers say that circumstances that frequently led to violence are familiar to us today, such as young men with group affiliations pursuing sex and alcohol during periods of leisure on the weekends. Weapons were never far away, and male honor had to be protected.
But I really, really love the way that these historian scientists have labeled these events. Alan stabs William from Devon in the neck. Oh, Alan. Oh, William.
And, some of these stories, too, are not just students attacking students.
Yeah.
There's one where 3 of the teachers attack a kid from a different region and murder him.
What?
There's
the teachers.
Yeah. There's infighting and it's just a blade. Anyway, this is, by the way, what you're seeing here is what the Florida school system will, if it's not already, will look like as they continue to do this. Everybody carry a gun everywhere, even on campus.
Yeah.
This is why, it's not guns that kill people. They're right. It's people with weapons, usually young men, who are not risk averse and very sensitive when it comes to their feelings and pride, going around killing people. And this is any place where you allow people to have all kinds of weapons at this age this has always been the case. Now, the solution to all of this, why this didn't destroy Britain, is because very soon after this period, where the murder rate was insanely high in the greatest university,
London had more murders, but it was per capita.
Per capita, per capita though, yeah.
Yeah. London had way more murders. I'm looking at that map right now. London was London.
So one thing that helped clean all of this up, the Black Death came shortly after. And wiped out so much of the population that, whether you were from anywhere, oh my gosh, it's good to see another human face. I haven't seen it. I've been walking for days and it's just been nothing but dead people. And finally, but this is
hours of fun.
Just in time for Halloween.
It is.
Medieval Murder Maps is full of stories of medieval murder for ridiculous reasons. And in ridiculous ways.
This is exciting. Thank you. This is medieval. It's amazing.
Yeah. What's interesting here too is that as you read the reports of who killed who and why it happened and who was responsible, you also have to keep in mind that none of it is true. Likely.
Who gets the records?
because they had juries of convenience back in those days, or judge panels of convenience that would more often than not try to come up with a reason why it was justified for the person with influence to have committed murder, and why it was completely a crime for this person who claims not to have been there to have committed the crime. Like, so there's going to be there's a very thick layer of whoever's writing this is probably doing so from a very influence at the time, medieval time kind of perspective. So
but they're taking whatever information is available to, and so whoever wrote the history in the first place is influencing what we know now.
Well, yeah. But the stories are still good, but also know that all the murderers are probably innocent. All the victims had it coming.
Yeah.
Well,
no,
maybe not.
Yeah.
Hang on. That can't always be true.
Hey, science. Last week we asked people to ask stupid questions. And I got a question. What do we think about the Skinwalker Ranch? My answer is I don't think about it.
Okay. You will have to tune in to the show 2 weeks ago when we talked about this.
You did talk about it. I didn't.
With Brian
you talked
Dunning.
with Brian. Honestly, though, my answer is I don't think about it. It's I don't even really. It's not in my zone of awareness.
So here's what Skinwalker Ranch is in a nutshell.
Oh. You can go and see UFO things and Bigfoot. You go there and they go. You might have an experience.
No.
But
so here's the real story. The real story is that the land was owned by somebody who got Harry Reid to give them a no-bid millions of dollars contract to study UFOs. Mm-hmm. And they collected that. They created a call center on the Skinwalker Ranch where they collected stories about when people would report UFOs.
Right.
And they were supposed to filter it up and analyze them and see which ones were real or whatever. There was never science fiction-y stuff happening there until that program ended and they needed funding and they started telling stories as though they were taking place on the ranch. But the ranch itself never had any form of supernatural anything about it. That's not why anything was brought there or done there. It was just where the guy owned land who got this federal funding from his buddy, who was the senator from Nevada, and that's the story. So it would be like this.
We don't need to talk more about it. We could say we don't think about it. It's fine.
No, I did think about it, and it turns out the whole and now it's a show where now it's a ghost hunter show because the same people. By the way, the same person who's I worked for the federal government actually worked for the Skinwalker Ranch guy who got this contract. He didn't work directly for the government when he was doing his UFO investigation nonsense for the government. It was government funded, but he was working for the Skinwalker Ranch guy. That's the Lozano guy who keeps coming out and "Oh, people knew about UFOs." So even that he wasn't part of the government. He was part of that.
But you talked about this 2 weeks ago. So
and we talked about it 2 weeks ago with Brian.
Yeah. So somebody was
and so we're doing a tight 90. So we need to wrap it up 20 minutes ago. So I have to get to the point where I describe time travel.
Oh God.
And then we can.
Wait. What? No, we're not.
We need to end the show.
No, I think we finished the show. We've done it, right?
Oh, I think we have.
These are all.
Good.
These are all our stories. There we go. Okay. Thank you, everyone, for joining us for this episode of This Week in Science. I do appreciate that you've stuck with us. If you stuck with us, I know some people had to go to sleep because we got going past our tight 90. But if you have your stupid questions, be sure to ask us, because why not? We'll answer your stupid questions. Let's go.
Also, I just want to make a quick correction. Eleanor is saying something out there about anti-vaxxers. So I want people to stop saying anti-vax and say pro-disease people, the pro-COVID people.
Yes, let's change it up. Yes, you're not anti-vax. You're just
the pro-whooping cough people.
You're just pro-disease spread.
The pro-pox, the pro-plague, the pro-pandemic folks, the ones who want a pandemic.
Yeah,
they're not anti-vax. They're pro-pandemic.
They're so propane. I can't even handle it. Okay.
And propane accessories.
In our accessories, I would like to give a shout out to everybody who's in our chat room right now. Thank you so much for being here and chatting. People in our Discord shared some great photos and images that I popped into our live video stream tonight, which was kind of fun to do. All of you really make this live show even liver. So thank you for doing that. Gord, Arnlore, others who were here who helped to kind of keep the peace and make sure that our chat rooms are nice places to be. Thank you for being here and for being those people. Fada, thank you so much for doing show notes. Thank you for doing the social media. And I know we need to sit and talk about social media stuff. I know social media is weird right now. Rachel, thank you for editing the show. And Identity 4, thank you so much for recording the show. And of course, can't go any further without thanking our Patreon sponsors, because we really
Patreon
can't do it without you. So thank you to Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, Rick Loveman, George Corrispiel, Pierre Velazarb, John Watners-Family, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Jonathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan Shubu, Sarah Forfar, Don Mundus, P.I.G., Stephen Alderan, Daryl Myshak, Stu Pollack, Andrew Swanson, Fredis 104, Sky Luke, Paul Runovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tank, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Remy Day, G. Burton, Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, Idaho Mojo, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Osters, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkon, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Summerly, Patrick Pecoraro, and Tony Steele. Thank you for all your support.
Wow. Look at handsome group of people that is.
And I do want to add more people to this list. There's a ... at the bottom of my list that I show here because I want to add more names. So if you are interested in supporting us on Patreon, please head over to twist.org, click on the Patreon link, and choose your level of support. We really can't do this without you. On next week's show,
we'll be back broadcasting live Wednesday, 8 p.m. Pacific time from our YouTube and Facebook channels and from twist.org/live.
Oh, yeah. And I wasn't ready for you to go there because usually you talk about 5 a.m. or whatever in European time. But if people want to listen to us as a podcast, they should really head over to wherever podcasts are found and look for This Week in Science, because we're there and you can subscribe. Get your friends to subscribe too.
Get your enemies to subscribe.
Get
as well.
Everyone.
Maybe
get your frenemies. How about your frenemies and your families and your dogs?
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org, where you can also sign up for a newsletter, which we probably aren't going to do because we spend so much extra time actually doing the show that the thing that we were going to talk about, we usually have already.
You can contact us directly. Email me, Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, Blair at blairbaz@twist.org, tell her that we're ready for her to come back whenever she's ready.
Beg her to come back. Inundate her with emails.
Just please.
And Twitter things and
we need
other
your
places.
animals.
Yeah, because I try, I brought like a self-amputating bug story, but that's her.
I know.
That's her.
I talked about cats. What am I supposed to attack?
Yeah.
Fluorescent cats. I don't know. Whatever you do, put TWIS in the subject line because otherwise it's going to get spam filtered into one of our poorly regulated memory neurons or even into, just some comfortable cat purr where, we go and nobody does anything because it's
Yeah. You can also hit us up on the platform formerly known as Twitter, which is now being represented by a very fascist-looking X symbol, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. At Jacksonfly probably isn't there anymore, though. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes during the night, please let us know.
Justin, I forgot to tell you, I got a book of periodic table haikus sent to me. It was to the KDVS, mailbox. Probably six.
How did you get it then?
No, it was probably from 6 years ago. And the new public affairs director was like, hey, we have a bunch of stuff for you. And
I bet they do, because we used to get 20 books a week or something ridiculous.
Yeah.
We used to just constantly, we'd come in there and there'd be a stack of new sciencey books, which was always a blast.
Now I have a stack of old sciencey books.
But if we never turned off the mailbox for how long has it been since we've been at KDVS? 15 years?
But we're still there. We're still there on the air.
We're still on it, but we are physically nowhere near the campus, which means there's a box somewhere in this poor, small, tight little recording studio, radio studio.
Just got mailed to me.
We keep getting, there's This Week in Science books. They keep on, what do we do with them? They've probably built a fort. There's probably guys a nice desk. Yeah, I made it out of science books for the This Week in Science people. Who
but there's a whole periodic table haiku book that I need to. I'll read some haikus from it for you. Anyway, we'll be back here again next week, and we hope that you'll join us again for more great science news.
Yes, we hope. And if you learned, anything from the show, it's that Justin has zero recall. It just merely fills in what he thinks should be the next logical thing to take place. What's the next logical thing? If you learned anything from in your head, remember. Is that what it is? Hang on. Let me go look up the right.
Remember. It's all.
If you've learned anything from the show, remember.
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.